At my company, Desktop Genetics, we work with a technology called CRISPR genome editing. And today I'd like to tell you about how it works, how researchers can use it in the lab, how doctors will soon be using it in patients, and what it means for our society and some of the decisions that we'll soon have to make as a species. So first, what is CRISPR? You can think of it as a find and replace tool for DNA. In the same place that you can press like Command F on a Word document and you can change one word to another, you can now do this with a DNA. You can change a sequence to another sequence in a living organism. It has two basic components. The first is the nuclease. This is a protein that cuts DNA. And the second component is a guide. This is basically a homing device. And you encode it with a specific sequence. It binds to the nuclease, but it also binds to the genomic DNA itself. So it acts as that homing device that guides the nuclease to its target DNA in an organism. Now, you can package these two separate components into DNA, and then you can also package that DNA into a virus. And you can use either of these methods to deliver CRISPR components directly into a cell. Once you've delivered it to the cell sees this DNA, and it effectively boots up these two programs, the guide and the nuclease, separately. And then they complex together inside of the cell. And then this happens. So that blue blob there, that's the nuclease protein. And this red piece here is the guide. And that has a variable sequence. And you can design that using computers and synthesize it chemically. Now, once inside the cell, this CRISPR complex looks for the DNA. It goes into the nuclease, and then it binds to the genomic DNA, and it sees if there's a match between the guide and the genomic DNA sequence. And if it's there, you will get a double-strand break. Both strands of the DNA are cut. Now, at this point, the cell knows that something is very, very wrong, and it attempts to repair this using its own internal repair mechanisms. And typically, it will make mistakes whilst doing this. And as a result, this gene will become scrambled or functionally knocked out. It's no longer there. And the cell does that to preserve the function upstream and downstream, but has to sacrifice that region. The other way that this repair process could work is if you introduce a separate piece of DNA that you've chemically synthesized that contains new sequences, something else, a change, a modification that you want to make. And this is how you can introduce a specific mutation or knock it in. And so researchers use CRISPR for this knockout and knock-in functionality to add and remove genes, to turn them on and off. And this is functional genomics, where you can turn on a gene or you can turn off a gene. And you don't have to do it just singly. You can do it to many genes at the same time in a highly parallelized fashion so that you can find out about how these genes interact with each other. You can also use CRISPR in another way, to generate model organisms or model cells. You can knock in specific mutations that you may find scientifically interesting, or you found them in patients that have certain diseases, and you can put those mutations into cells so that you can study how this disease progresses. Or you can make a custom animal model, a mouse, a fruit fly, a monkey. The potential is endless. But what can this do for you beyond research? Well, simply put, you can use CRISPR technology as a cell therapy. You can take cells out of a living patient, and then you can modify them as you desire, and then put them back inside of the patient. Now, right now, there are studies going on, clinical trials, to do this very same thing. One example would be immunotherapy, where you would take the immune cells, white blood cells from your body, and you would modify them to recognize cancers inside of you as foreign. So you're really leveraging your own immune system to go after something which shouldn't be there, which it doesn't do naturally. Alternatively, you can use a gene therapy, where you're delivering CRISPRs directly into the cells themselves, into the tissues of a living patient. Now, in this case, you would probably package the CRISPR system into a virus, and then infuse this into a patient. One example would be to target the genes responsible for causing cystic fibrosis when they're defective. So you could literally repair the defective gene inside of the living patient. So how accurate is this process? How well does it work? Well, think of it as shots on target. Once CRISPR has entered the cells, it is working, not just on where you want it to go, but it can also target in these other places. And these are called off-target effects. And it's very difficult to control for this, but you can predict this with varying degrees of accuracy, depending on what computer models that you use. And every day that goes by, we learn more and more about how to predict this successfully. But ideally, you want to avoid off-target effects, because you don't want to accidentally knock out or scramble an essential gene. You don't want to get rid of something that is already doing a good job. You only want to hit that specific on-target location. The problem is all of this targeting is sequence-dependent, and it's complicated by the fact that we all have about 5 million genetic differences with the person sitting next to us. That's what makes us unique, but it also means that it's very difficult to design CRISPR therapies for individual patients. There's no clear-cut process just yet. So let's look at an example. Say I have 3 guides that will target an on-target region in patient 1. Right? And all 3 of those will hit successfully. If I take those same guides and I want to use them for the same disease in patient 2, 2 outcomes could occur. Either I may get no on-target hits, in which case it's quite a good case that the disease doesn't get treated. The patient continues to live, nothing bad happens, we can try again. The worst case scenario is we accidentally cut in some of their tumor suppressor genes, and we switch those off, and as a result, the patient could get cancer. So this is the off-target problem. It's very complex. And so we really need to be moving towards personalized genome editing. And there's 2 ways that we can do that. There's 2 processes that we can use. The first is to sequence the whole genome of the patient, where we will take some cells from a patient, and then we'll look at their DNA, and we'll build a very accurate digital model of what that looks like. The second thing to do would be to look at historical CRISPR outcomes, to analyze all of the data from how CRISPR behaves inside cells in humans, in animals, and other organisms, so that we can build a good model about how it will behave in you. Now, obtaining this data is relatively easy. This is some of our data from desktop genetics, and we see that there's about 152 countries that are using the technology. And every day that goes by, they're creating more data about how CRISPR systems behave that can be analyzed to build that predictive model. And that's what we're doing at my company, Desktop Genetics. We are building a machine learning system that can take both the whole genome sequence of the patient and all of this historical data and can design around the off-target problem. This is the vision of personalized CRISPR therapy. You go to the doctor, you're diagnosed with an illness, they take a sample from you, they sequence your whole genome, you come back a few weeks later, they've analyzed this alongside the historical data of CRISPR outcomes, and the artificial intelligence system has designed a unique therapy. Only you will receive this therapy. It won't take $1.2 to $2.5 billion to generate a new drug. It won't take 10 to 20 years. It could be done relatively quickly. But there's a different number, and it's possibly a little more scary. 1, you are both the patient receiving the therapy, but you are also the sole participant in the clinical study. Would you trust this process? Would you be willing to be the first to try it? I would, but only if it was sufficiently regulated. And so I believe that these kind of design tools, whether they're AI or otherwise, or whatever they will be in the future, should themselves be regulated before they can be used in the clinic. Can such CRISPR therapies really cure everything? I've talked about gene therapies and cell therapies, but there's an issue, which is that there's about 37.2 trillion cells in an adult human body. And you can't easily edit every single one of them, as you would need to in order to treat certain diseases. It would be very difficult to do this. So really, we have to do something else. We have to find a way to treat all of the cells at the same time, or possibly just to treat one. And so this would be editing the embryo at the single cell stage, as it then divides into 2 and 4 and 8. And 9 months later, when that individual is born, those changes that you might have introduced to the embryo would be present. And not only would they be healthier, if that's what you intended, but they would also be able to pass those changes on directly to their offspring. This is a scary prospect for most, and I'll discuss why later. But let's say we wanted to do this. How would this work? What would we be looking at? This is a technique called pre-implantation genetic diagnosis. This is a technique which in the UK is very, very heavily regulated, where essentially you cleave off a single cell from the 8-cell embryo, and you use that to sequence it. And from there, you build that accurate digital model, which you could potentially use to input into the AI system if you were so determined. But typically, how this works is that this is only given to patients or parents who are undergoing an IVF program. And the results are a report of about 419 different genes, or 419 different diseases, which could impact the embryo or the child when they're born. Parents receive this report, and they're able to make an informed choice about which embryos to implant and to carry forward in pregnancy. Typically, PGD is appropriate. It works. It allows parents to ensure that their child will not have a disease. But let me ask you a question. Let's say you were doing this with your partner, and you discovered that all of the embryos were to have a disease. Would you cast them aside and undergo additional IVF therapies, rounds and rounds, if it needed? Or, given that the technology is there, would you want to repair them directly? And I would note that the use of genome editing technologies in human embryos is largely illegal in most countries. Some countries have special dispensation for research purposes. But it's an interesting question that we must confront now that we're there and we have this technology. And while you're at it, if you're able to modify a disease variant and turn it into something neutral, can you turn a neutral variant into something advantageous and protective? Well, you can, or you could. These are some protective variants which have come out of various research programs, and they guard against certain diseases. Low coronary disease, low Alzheimer's, low rates of cancer, low type 2 diabetes. Ask yourself, given the choice, would you want your children to have resistance to these diseases? The ones I find particularly interesting are the ones at the top, LRP5 and MSTM, extra strong bones and lean muscles. Individuals that would have these mutations, which do occur naturally, would be able to resist osteoporosis and muscle wasting later in life. But then the question is, when does prevention or cure become human augmentation? Let's take an example. Let's look at lean muscles and osteoporosis. In the gentleman on the left, these mutations would allow him to be skateboarding well into his 70s and 80s. He wouldn't get osteoporosis. We would prevent muscle wasting. But those same mutations in the very same individual, when that person is younger, would also give them possibly an unfair advantage. They might make an excellent college football player and quarterback. But what about individuals that have these naturally? Do they have an unfair advantage? It's not really clear, and it's very, very difficult for us to assess what is prevention, what's a legitimate medical intervention, be this kind of human augmentation. Now, in modern medicine, we say that prevention is the best cure. But how far are we willing to go in order to uphold this ideal in the age of genome editing? When vaccination was first invented, people said that it was ungodly, that it was unchristian. They said that it was a violation of human rights, particularly because states implemented programs where vaccination was mandatory. Now we think very little of vaccination. Well, some of us think a lot of vaccination and are opposed to it. But I would ask the question, is the right to healthcare equivalent to the right to genome editing, whether it's cell therapy and gene therapy, or if it's editing the embryo? I don't have the answers, but I think it's important that we begin this discussion as soon as possible. And this discussion is ongoing. There are many meetings where scientists, politicians, policymakers, and so on have such discussions. Now, with regard to human rights, there's 175 signatories and 169 states that have ratified the International Covenant on Social, Economic, and Cultural Rights. And in this international treaty, they have agreed to be obligated to recognize the right of everyone to the enjoyment of the highest attainable standard of physical and mental health. How might states interpret this now that we have genome editing available? Will states, or individuals even, consider these two things equivalent? Would you say that they're equivalent today? Do you think people 100 years or 200 years from now would agree with that? And given the choice, as I asked, would you want your children to be protected against certain traits or diseases? Or we could go a different way. There is a group in the United States, the Council for Responsible Genetics, that have proposed a genetic bill of rights, which says the opposite. All people have the right to have been conceived, gestated, and born without genetic manipulation. Which of these do you think is more accurate? What reflects your choice? Regardless of your answer, which may or may not demonstrate a consumer will for genetic augmentation, it's important that we understand a little bit more about what we're learning about disease and protective benefits, protective gene variants. In the UK, Genomics England is an organization which is sequencing the genomes of 70,000 patients that have rare diseases and cancer. It's also sequencing the genomes of their families. Now, the outcomes of this will be populations, associations of variants with specific diseases, and then we could make causal or correlative links. This would have profound implications for early diagnosis, and it would help guide the development of cell and gene therapies. Techniques which can be used to elucidate disease could also be used to discover more complex links between genetics and human behaviors and characteristics. In China, the BGI Cognitive Genomics Group is assessing the genetics of cognition. It's looking at the impact of the environment on personality. But this is a really ethically challenging area and really challenges some of our deepest core beliefs about nature and nurture. What if they do find these links? The issue is that states, just as individuals, have divergent views on what is ethically sound to research and what is appropriate to prioritize in terms of their technological development, as well as the development of their human capital. What would it look like if a state decided to put psychopathy, if there was a genetic link, onto one of these screening lists? When would a normal brain or normal personality end? And when would a psychopath's begin? How could you tell? Many of these complex human behaviors and characteristics are on a spectrum, and it's difficult for us to assess when these things start and stop. What might an authoritarian state do if it implemented a research program to look at the links between genetics and obedience to authority? Once again, we see that there are challenges to our ethics, nature and nurture. But what happens if a state decides to go? Nothing too complex, but they implement a program to improve some aspect of healthcare. How would other states respond? What would happen in that genetic equivalent of the space race? And how do we prevent it? I would like to see states come together to have these discussions of what is right and wrong, what is acceptable. I don't know what those restrictions are, and it will be very difficult for us to come together as a consensus, but we must try. My concern is that states and individuals that have both the financial and the technological means will choose to give their children or their offspring, their populations, strategic advantages. And we can't easily tell how different states or people will determine what a strategic advantage is. There is a risk that because we do not have a discussion about human rights to genome editing, it could be abused. And equally, there is a risk that any single implementation or use of this technology will set off a chain of events that destabilize social mobility and the international order. I believe that cell and gene therapy is a tremendous opportunity for patients and for the medical field that we should embrace. But I acknowledge that therapeutic CRISPR is incredibly complex. It must be designed around the patient, and the design tools themselves must either be regulated or form a core part of the regulatory process. I believe that states must come together, and they should reach a consensus as best that they can. And that will be difficult, but it's our responsibility to make that happen. I believe that these conventions should reflect today's society. Again, very difficult given that we can't agree on how to regulate genetically modified organisms, let alone humans. But I also believe that these conventions should be updatable. We should provide our descendants with the mechanisms to change the decisions that we make today or in 30 years or 100 years from now. And there should be regular reviews, just as there are in other conventions. Everyone should be involved. And I think that it's our duty to have a discussion with our friends, our families, our politicians, anyone that we can. And in order for you to do that, you must learn. And so I would invite you all to email TEDx@deskgen.com, and you'll receive an interactive version of these slides with all of the sources, some great books, fantastic review papers, some of the fundamental research principles, and many other things. So I'd like to thank you for your time. Thank you.